want to thank everybody for coming today. And the talk we have today, sorry, I'm just letting some other people in. The talk we're having today is by a PhD student at University of Massachusetts School for the Environment, and Evan Craig. He is actually one of my PhD students. We started working together when we were both at Antioch University, New England, and then I transferred away from Antioch to left that position, and I've been full-time at University of Rwanda, but I have an affiliation with UMass Boston School for the Environment. So Evan transferred there and we continue to work together. And I'm really excited to see the work that he's going to be presenting today. He's been working with small mammals, and he works with the Chicago Field Museum. So I definitely think I can see Alexis is here, and Alexis works with our zoological collections and probably is going to be really interested in this talk. And I know, unfortunately, Method Majamberi, who also is studying small mammals, is in the field. And I was hoping some of the folks from Karasoke Research Center would come because I know they're doing some work in this area. But we're recording it. I believe Emmanuel is going to be recording the talk. Actually, I don't, yeah, Emmanuel's here. So then it will be available for other people to listen to. So Evan, I'm going to hand it over to you. And so Evan is going to tell us about one of the chapters in his PhD dissertation, and this is one aspect of the research that he's been doing. So thank you, Evan. And with that, I'm going to mute and turn my video off, and you can take it away.
Okay, thanks for that. Yeah, so thanks for coming and logging on and listening to this talk. I gave it a couple days ago at the American Society of Mammalogists meeting in Tucson, Arizona, and now I'm on my way back home and I'm going to give it again. But one thing that happened is the actual presentation and everything that I had planned before that meeting is on this computer, but this computer is now just a paperweight. So if at any point during this presentation you have a question, or if the slides aren't looking right or something, just interject, and please bring that up or correct me because I'm using different software, and it went all right a couple days ago, but just a forewarning. So yeah, today I'm going to share my screen and talk about something that I might have found in the Simien Mountains of Ethiopia. Let's see. You can see the whole screen now, right?
It's not, oh yes, now it's in presentation mode, yes.
Okay, all right, looks like it's going to be good. Okay, so this is kind of preliminary. I've looked at the data a bit now, but I would still probably say that a better title for this is High Elevation Specialist Mammal Species Weigh More Than Their Relatives Down the Mountain, I think, because I've just done kind of back of the napkin or very preliminary analyses looking at a pattern that I potentially found in the Simien Mountains. But there we go. Before that, I should do a little bit more of an introduction about the process and about myself at the same time. So my name is Evan, and I'm from Wisconsin, which is one of the more northern states in the United States. We are typically known as the cheeseheads. It's because of our football team. Whenever you watch our football team on the national broadcast or whatever, all of the fans from Wisconsin have cheese on their heads, and that's a whole different story. And you can ask me, maybe we'll talk about that later, but we're known for putting cheese on our heads when we watch football. Anyways, last week I was in Arizona, which is a lot further south, and it was a lot different climate than what I was used to. And I'm sure a lot of people or, on the street don't think about their surface to volume ratio. But when I was in Arizona last week, I was definitely thinking about my surface to volume ratio. And maybe I had a little bit too much volume and not enough surface. And the reason for this is can be, I guess, explained by Bergmann's rule, which we will get into, and I think is maybe an underlying pattern behind what we are finding in the mountains with respect to elevation and the distribution of small mammals with respect to body mass. So when I was in Arizona last week, I was thinking, if I wanted to get back to a climate that was more comfortable for me, what would I have to do? Well, I could go back north, like where my state is, and if I went to the Canadian border, which is about 1,850 kilometers, I would be more comfortable. Or I could just go up 1 kilometer, any of the mountains that surrounded Tucson, the city that I was in. Going up 1 kilometer would give me effectively and theoretically the same 7-degree Celsius change in temperature. So this Bergmann's rule is generally thought of as animals found in colder climates tend to be larger than those found in warmer climates. And it's been taken in a bunch of different directions. A lot of studies have interpreted colder climates as latitude, higher latitudes. And it's recently been applied, I think, more intraspecifically. So an example of that might be here in the US, at least, we have grizzly bears in Montana, which is in the 48 states. And then we also have grizzly bears in Alaska, which are at much higher latitudes. And you might say, well, Bergmann's rule might be the underlying mechanism or principle behind why the grizzly bears in Alaska, way up north, are larger than the individuals that are in the 48 states, despite them being the same species. But Bergmann actually introduced this back in the day in terms of closely related species, or interspecifically. So Bergmann actually said, if we could find 2 species of homeothermic animals, which would only differ from each other with respect to size. So if you have two species, yeah, and the only thing that's different is size. Everything else seems to be the same in this theoretical example. The geographical distribution of the 2 species would have to be determined by their size, because that's the only thing that's different between these. If there are genera in which the species differ only in size, the smaller species would demand a warmer climate to the exact extent of the size difference. So this was Carl Bergmann's thought. And I was thinking about this thought when I was working with some data from field work in Ethiopia in 2015. We went to the Simien Mountains and we surveyed small mammals at 4 different elevations along the elevational gradient of one of the sides, the northern aspect of the mountain. And I started looking at the data, and there were 5 congener pairs that we found, so 10 species in total, 10 species that had a relative on the mountain. And I started looking at the data and seeing where they would sort out elevationally. And I started to look at a couple of the species, or a few of the species actually. And what you would see here is these are 4 different species, but of course they're of 2 different genera, right? There's 2 genuses here. Obviously, the ones with 5 feet probably are the same genus, but their colors show you that they're different species. And it started to look like this pattern was holding up. The larger species of the related pair was higher than the other one. So sometimes, in science, or maybe this is a young scientist or fresh into the field kind of thing, but you start to look at data and it starts to form a pattern, and then you keep adding more data and you get really hopeful. And I kept adding more data, and sure enough, they all conformed to this same pattern, where the higher of the two related species is the bigger one, or had more body mass. In this instance, I was using average weight of that species, until I got to the shrews. And the shrews didn't work out. I think that's very predictable for shrews because they aren't very predictable. Yes, is there a question? No? Okay. So the shrews didn't work, but that's okay. Maybe I'll just revise my hypothesis to look at the rodents for now and maybe tackle shrews later. So I wanted to graph this to get a better visual indication of what might be going on. So the next few slides are going to be based on this kind of template that you see on your screen. Weight is the x-axis. So as you go farther away from the y-axis, it's getting heavier in the weight ranges. Elevation is the y-axis. So these lines that you see on the graph, the length of the line shows the elevational range that a species was found at. So each line is a species, and the colors are the genera, just like in the example I used with the animated rodents. So this would show two species belonging to the same genus, and that's color-coded as green. And the length of these, like I said before, on the y-axis shows what span they had elevationally for their range. So let's go through this first Simien Mountain example, because this is where I started to compile and play with the data and see if it fit. This is Stenocephalemys in Ethiopia, and there's two species on this mountain, as you can see. And the first one has a range that goes to about 3,400 meters, okay? And the second one goes just a little bit above 4,000. And as we start looking at these graphs, what I would like to see, or if you'd like to see the pattern, you are looking for this same kind of instance where the species furthest to the right on the x-axis is higher than the one that precedes it. So here's another example from Simien Mountains, Dendromus, those two species. And then finally, with all of the species that we found on the mountain, minus the shrews, these are the rodents. So everything seems to work out. Then I went to the other big Ethiopian massif, I guess. There's two that are really the claim to fame there. There's the Simien Mountains and the Bale Mountains. The Simien Mountains are in the north of the Rift Valley, and the Simiens are in the north of the Rift Valley, and the Bale Mountains are south. I started looking at the data here, and it started to line up in the same way, with the higher species on the mountain being larger than the one further down the mountain. And there's even an instance here where there's 3 different species of rodent belonging to the same genus, and all 3 of them conform to this same pattern. The Bale Mountains are actually pretty remarkable in this regard, because there's a lot of congeners on the mountain, and each time it actually does conform to this pattern. So to see if this was maybe by chance, or maybe even just Ethiopian, because Ethiopia has a lot of endemic species, and maybe the way the endemic species behave there is different than in other places, just due to their endemic nature. And so I looked at other mountains in Eastern Africa, in the Rwenzori Mountains. I found the same pattern, although it's starting to get a little bit harder to discern where the elevational distributions actually begin and end. Because if you have these lines, like the green and blue lines that span the entire elevational range, one confounding factor when you go to analyze the data is not knowing if they actually do exist above or below those minimum and maximum elevations, because they weren't ever sampled. Mount Kilimanjaro has only 2 pairs of congener species, And this is one of the exceptions. You can see on the right, the Otomys species. This does not conform to the pattern that we would expect. But I will say, just as a side note, is the larger of these two Otomys species here, the one that doesn't go as high in elevation, that is only based off of one specimen, where all of these other species are averages of all of the specimens that were caught on that during that survey. This is just represented by the one. So there's not a lot of confidence in that result. Mount Kenya, there's two slopes on Mount Kenya, the Chogoria slope, and this one shows a couple different instances that I'd like to highlight because it comes into factor when analyzing any possible pattern or testing the data. So for example, if you look right in the middle of the screen, the Graphirius genus, you have two species, okay, and they're one is heavier than the other. They're different in terms of their body mass. And one of them does indeed extend higher in elevation, and that's the larger one. But you'll notice that the larger one also extends lower in elevation than the smaller one. So this would be one of a prime example of how it's difficult to test this hypothesis before actually defining the question of, defining the concept of what is higher, right? Because a higher elevational range could be the midpoint of their elevational range. It could be, are you comparing their maximum elevations for each species? Because in this example, if you were comparing the maximum, then yes, the heavier species would be higher. But if you're comparing based on the midpoint, actually, it probably wouldn't be higher because the midpoint of the other species ends up being higher on the graph. But again, we don't know where that second species lower limit actually is because that's where the surveying just started. But I think it's a good example of defining the elevational ranges that needs to happen before this can actually be statistically tested. So these are just very preliminary numbers that I was trying to jot down after looking at the data. And I think it has three of the most basic ways that you could assess this pattern. So the first one is max elevations. That's just comparing between the maximum, the top of those ranges for each of those species. And if you look at the top of the ranges, the max elevation of the heavier species is greater or equal to the max elevation of the lighter species. And that's true 94.7% of the time. If you do the opposite and you look at the minimums are going to tell you which one is displaced elevationally on the mountain with respect to the other. The minimum elevation of lighter species is less than or equal to the minimum elevation of heavier species. That's 95% of the time true. And then finally, if you were to look at the midpoints, the midpoint of the heavier species being greater than the elevational midpoint of the lighter species is 94.7%. It agrees 94.7% of the time with the data that I used. But I did put an asterisk there on the elevational midpoint assessment because you would actually have to know the minimum and maximum, the true minimum and maximum for each species for that to have any real merit. So some limitations that I've run into while trying to look at this data and discern this pattern is distinguishing non-adult individuals when it's not explicitly recorded. So let me explain what I mean by that. The only mountain data that I used here that was actually my own was the Simien Mountains. So I knew all of that data and I knew the records and what was recorded and how I recorded it, and maybe if I made abbreviations in my field notes, things like that. But all the other mountains were offered from colleagues or in one instance, a colleague that had passed away, and I get the records. And if I see 10 individuals of a species, and most of them are 100 grams, if I see one that is 51 grams, and its length measurements for its ears and its legs and its body length are all, it's an outlier. How do I know if that is a adult individual or not, in terms of methodologically supporting my decision for making that decision? And the reason I want to do that is if I'm comparing between average metrics or average body size, body mass, maybe I would use body length, any of these, I want to average between anything or adults only and nothing that's not an adult. So in that instance, I wouldn't know where to draw the line between this is just an outlier, this is a very small adult, or this is actually not an adult if it wasn't recorded by the scientists in the field. So the age is one limitation, and then reproductive and social status. Sometimes this is recorded, sometimes it's not. Sometimes this is known, sometimes it's not, just like the pathogens and parasite loads when the data is being recorded. There could be, parasite loads at that time of the year, seasonality or something like that that was occurring that would affect the sampling and the trapping, and that's a potential confounder that needs to be addressed. Different trapping protocols detect some better than others. This is very true with small mammals. It's especially true if at a later point I'm going to start considering shrews and maybe looking at why shrews don't conform to this pattern, because shrews are known for being undetectable if you don't use specific traps catered to them. For example, we found a new species of shrew in the Simien Mountains simply because we used pitfall buckets this time when everybody in the past had only used snap traps or live traps, bait traps. And of course, a big limitation of this kind of thing, Bergmann's rule is an ecogeographical rule, and all ecogeographical rules share this suite of environmental confounders. For example, if you're comparing between two different latitudes of the grizzly bear example that we were talking about, latitude is really just a proxy for climate, right? So your assumption would be that it's the difference in climate between these two latitudes that account for this difference. And a lot of times that's thought of in terms of heat, but there's a lot of other things that change in latitude. There's humidity, and there's going to be changes in elevation, and elevation actually, as we saw from the example in the beginning, has a much stronger effect on temperature and climate, and a lot of these environmental scales. So for example, if I were to go from Arizona in the United States to the Canadian border, in the inset picture, you can see that line. This would be the elevational profile along that route. So if I was comparing between a point at the beginning and maybe a latitude midway through, it would be, what would be the difference here, like 3,000, something like that. It potentially could be a difference of 3,000 meters, which would work out to 21 degrees Celsius difference, just because of the elevational difference, when really you were trying to compare between latitudes. So another way I went about this, and I'm starting to explore, is to take the log, or the ratio of log abundance of the big and small species on each of the mountains individually. So this is the Simien Mountains, and basically what this is taking the abundance of each congener pair. I would take each pair of related species, and the big one, I would document the abundance, and the small one, I would document the abundance for each of them at each of the 4 elevations we surveyed. And then I would take the log of those numbers and create this ratio, because what we would be looking for in the end is for the resulting line to be increasing at any degree. The red dotted line is what you would see if there was no underlying pattern detected. That would mean that the big ones are not any more higher than the small ones. Potential explanations for why the shrews don't conform to this pattern. In reality, my result, I figured out, shouldn't be very surprising because shrews are kind of known to not conform to Bergmann's rule as it is defined by latitude. So doing it by elevation, one might expect that the same would occur. And I guess it's also not surprising because shrews have very complicated or I don't know, maybe advanced physiological adaptations. For example, some of the highest living mammals on the planet, the highest elevations, are the smallest shrews. And typically the large shrews are the ones that you find in the more lowland tropical habitats. The new species of shrew that we found in the Simien Mountains was at 4,000 meters, and its body mass was only 3 grams. That's like a hummingbird. Thanks. So that was my intro to the talk, and I want to thank you for coming and logging on and listening to it. I want to thank, of course, Beth and these institutions that you see here. I get a lot of help from folks at the American Society of Mammalogists, my own institution, UMass Boston. The Field Museum is who I worked with to actually conduct this initial research. And the gentleman that you see in the picture with the cheladas in the Simien Mountains was the collection manager at the Field Museum who made this whole research expedition possible in the first place. And with that, I'll take any questions or comments that you might have.
Oh, great. Thanks so much, Evan, for that talk. And I think people are clapping in the background uproariously.
I'll hear it.
So let's see. I don't see any comments in the chat right now. So I want to invite people. You can put it into chat. Some people, if you don't feel comfortable talking directly or your microphone isn't working, and otherwise you can unmute and make a comment or ask your question. And while we're waiting for some questions or comments, I just wanted to ask, are you in the data sets able to control for effort? I was just wondering, the different elevations, if the effort was similar across the different sites, because I imagine you did mention like different traps, and how they may capture differently, but I was just wondering, like trap nights or days or something like that might have an impact. I don't know if you handle that in the analyses.
So far, I haven't accounted for that. I've been using some reasoning that I used when I looked at when I compared distributions between the Simien Mountains and an older, like 88 years ago data set. And when I did that, I realized that the methodologies are very different and can only really be interpreted in one way. And that means that, for example, since we went to the Simien Mountains with a standardized sampling regime, and we had a standardized trap effort, we spent like 7 days at each elevation, did thousands of trap nights. If they found a species at an elevation 88 years ago, but we didn't, we can be fairly sure that species wasn't there. But if we found it now and they didn't find it 88 years ago, you can't say the same thing because you don't know their trapping effort. And I think I kind of took the same, or I've been taking the same approach as far as looking at this data, where just using what we know as far as it has been documented in these ranges and using that for this preliminary analysis or look at the data. But I'm trying to figure out some how I could implement like LME4 or some generalized mixed modeling to look at all of these mountains in Eastern Africa that would account for those types of residuals like trapping effort, or even the latitudinal or longitudinal distance between the mountains, and if that has any effect. Yeah, did that answer your question?
Yeah, thank you. Thank you. So there's another question here in the chat from Emmanuel Dushimimana. He was asking, what do you consider for concluding species at high elevation have a higher weight compared to the ones at low elevation? I think he means like the reason why that is the case.
Yeah, that's a good question. I've kind of been making myself be patient with trying to answer that question in too much detail at this point, because I'm first trying to like figure out if the pattern holds up, and then if the pattern does continue to hold up, I'm going to, you know, embark on that aspect of, okay, so what is the mechanism behind this pattern? But it is a good question, because that's a lot of the criticism that ecogeographical rules or Bergmann's rule or Allen's rule gets from folks, is, you know, there's a lot of exceptions to Bergmann's rule. They can point to these papers or these surveys that these rules don't actually hold up. But a lot of times I think that we're getting lost in the weeds when we do that, because the point is that Bergmann's rule and Allen's rule and these ecogeographical rules, the point I think is that they conform more often than they don't, which indicates there is something going on, and it might not be heat or you know, it might not be temperature or humidity or, you know, just one of these mechanisms, but many of them combined. So I hesitate to answer that yet, but my assumption would that it be related to either physiological processes having to be more volume and less surface area at higher elevations where it's cold, for regulating body temperature and heat. But I can also see, you know, very plausible that it could be energy resources, food resources. Shrews are insectivores and rodents are granivores, or, you know, they're grazers, or they eat seeds. And how is that different at the top of the mountain compared to at the bottom? If you're an insectivore, are the bugs bigger? The higher you go on the mountain, are they smaller? Are there more big bugs? You know, so I could also see the resource availability playing into this as well.
Yeah, thank you. I wanted to see if Emmanuel, if that answered your question, or you had any thoughts on why that pattern exists.
Hello. Hello?
Yeah, go ahead.
Hi, Emmanuel.
Hi. Yeah, I would like to ask you if you consider for one species or different species, because as we know, each species have some different species. Each species have to adapt to its habitat.
Okay. Are you asking like intraspecifically if I looked at the same species higher and lower, and if there was a difference?
Or maybe Emmanuel, if you want to put it into a chat so that we can read it, that might be helpful.
Well, in the event that was the question, I will address that. I did look at within the same species at the minimum and maximum of their elevational range. I didn't go as in depth as I did with this intraspecific, like, I don't want to call it analysis yet because it's quite preliminary, but I did look, and there seemed to be, a couple species that were a little bit larger on average, the higher they were surveyed or collected on the mountain, but it wasn't something that was an obvious pattern across the board.
All right. Well, there is another comment here from Alexis. He said, I would like to ask the method used to identify species, because there are some species which are more difficult to identify, such as moose and Dendromus species. And according to your observation, is it seasonal effect, the distribution of the small mammals? Is there a seasonal effect? So those were the two questions from Alexis.
Really good questions. Yeah, so we identified the species for the Simien Mountains, the one that the data was based on, that I started looking at the Field Museum in Chicago. So after we collected a bunch of specimens, we took them back to the lab. Skulls were extracted, and we were able to do a lot of morphometric analysis with them until we're quite sure. In fact, I don't have any doubts about the identifications, but it's a very good question, because a lot of times if you pull survey data, small mammal survey data from another expedition or something like that, a lot of the times they might be field identified, and you're totally right that field identifications can be very problematic, because there are many things in the field that are very difficult. And I think that, people that study shrews would actually say impossible to identify or discern in the field. So for us, we used many different methods to identify our species, and where phenotypically or morphologically it wasn't straightforward, we did rely on Site B DNA analysis. Oh, and the seasonal distribution, that's also an excellent question. But the surveys that we did, or that I've used here, are single trapping events. None of these are like collected over a series of expeditions. These would be going to a mountain, staying there 3 or 4 weeks, going to different elevations, and coming home with the data.
All right, we have time for a couple more questions. So Method posted. So Method is also he's a research associate in the center, and he's an academic staff in biology department in the College of Science and Technology. And he said, I would have asked a number of questions, but he has limited connectivity. I know he's coming back from the field right now. I like your study. I would like to welcome you for collaboration, both with me and my students. I have some of them I've been training on terrestrial small mammals. I have a PhD work on taxonomy, diversity, and habitat use by terrestrial small mammals in Nyungwe, and he's going to start field work in July.
Great, thanks, Method.
And Method, I should say that, Julian Kurbis has done work on small mammals in Nyungwe, and Julian Kurbis is on Evan Craig's committee. He is a researcher at the Field Museum in Chicago, and one of the 3 committee members. It's 3 on your committee, right, Evan? I can't remember. Yeah, good.
Yep. Okay.
Any other questions or comments? I know there's several other PhD students I can see here in the group. I don't know if any of you have any questions for Evan. And if not, well, I think we can end there. And Evan, we will invite you back when you have other parts of your PhD studies done, because I know you've got several other chapters planned. So we thank you very much for taking time to come and talk with us. I do hope that you and Methode could develop a collaboration. We'd love to welcome you back here. I know you visited here before, and now there's quite a bit being done with the small mammal collection. So yeah, we hope that maybe there'll be an opportunity to collaborate. And thanks to everybody who attended. And do feel free to propose a talk if you have a talk you want to give, or if you would like to propose somebody who could give a talk, then we would love to welcome them. And Vinat is the one who organizes those. So yeah, I saw Emmanuel just posted this Dushimimana, Emmanuel. He was saying, we know different species can adapt differently at habitats by considering adaptation. One species can have more weight at low elevation. Yeah, so he was asking for within species or an interspecies variation in weight. So if that would, how that fits into your model here.
Yeah, so the same species, we didn't find a lot of, or I guess I didn't find a lot of change along the elevational cline. And that's a good question, because I think one thing that I forgot to mention was the reason that I was looking at this interspecifically. And I think the reason that Bergmann might have presented the idea is because if you're thinking about a speciation process, if you look interspecifically, you're really looking at what's happening after these two species became new species, and the way that they're starting to diverge from each other. And I think that that's a really interesting way to look at the differences in elevational or any environmental gradient, whether it's elevational or otherwise.
Great. Okay. Yeah, I'm just seeing. Prospero is asking, is there any correlation between body weight and species diversity if those parameters were considered? I'm not sure what that means, but
yeah, I'm not sure the question. We did consider species diversity both at the sites and on the mountain as a whole.
So the graphs showed different species and their body weights. So yeah, I'm not sure. Maybe Prosper, do you want to try to expand on your question or what you mean?
Yeah.
Well, I'm not sure. I know Prosper was, I don't know, Prosper, are you able to unmute and explain your question a little bit more? Okay. I don't know if you want to try to address any aspect of that before we close today, Evan?
Yeah, I guess I didn't look at a correlation specifically between body weight and species diversity, but yeah, I'd be interested in what the
yeah, maybe he means like if there's more species at a certain elevation, would you have any predictions about what body mass would look like? I'm not really sure what where that question was headed, but maybe. All right. So I don't see any other comments in the chat, and I don't see that anybody has their hand up. So yeah, thank you all. I hope you have a great afternoon. Thanks for attending, and look out for the talk next week at the same time, same place. Thank you. Bye-bye.
Thank you.